All right, if everyone can take their seats. That was quick. I didn't even need Harriet to whistle, right? Well, welcome everybody to the 2018 Brains and Behavior Retreat. I'm Dan Cox. I'll be your MC. So just a couple of words real quick for the speakers. To keep us on time, you'll see me stand up at 15 minutes in. And then I'll give you this when you have 2 minutes left. So I'm going to be pretty strict about staying on time. We have a really exciting group of talks this afternoon from, as Nancy mentioned, many of our representative departments, so I'm looking forward to this lineup. Our first speaker is Jonathan Borland from Elliott Alber's lab, and he will be talking to us today about sex-dependent regulation of social reward by oxytocin. Welcome, Jonathan.
So thank you, Dan, and thank everyone for being here today. And so yes, I'm Jonathan Borland. I'm in Dr. Elliott Alber's lab in the Neuroscience Institute. And today I'm going to be giving a talk that looks at how the rewarding properties of social interaction may be more rewarding in females compared to males, and how oxytocin may have sex-dependent effects on the social reward processing. So there are well-known sex differences in the instance of many psychiatric disorders. For example, PTSD and depression are more likely to be diagnosed in females. However, autism spectrum disorder is 4 to 8 times more likely to be diagnosed in men compared to women. Now, one prevailing theory is that differences in the neurochemistry between men and women results in sex differences in the susceptibility and predisposition for psychiatric disorders. Now, historically, the rewarding properties of various drugs of abuse, sex, and other things such as exercise and food have been well studied. However, the rewarding properties of social interaction are not as well known. Thus, as a graduate student, I'm particularly interested in understanding sex differences in the rewarding properties of social interaction, and specifically sex differences and the neural mechanisms underlying social reward. Now, for clarity, from an operational standpoint, reward is something that, when delivered, increases the probability of a behavior. It reinforces the behaviors associated with the reward. Thus, the rewarding properties of social interaction are critical for the expression of adaptive social behaviors and the development and maintenance of beneficial social relationships. Furthermore, deficits in social reward are associated with many psychiatric disorders, including autism spectrum disorder and schizophrenia. Now, Syrian hamsters are an excellent model for investigating sex differences in social reward, mainly because both males and females display highly robust dominant-subordinate relationships, behaviors also seen in primates, and behaviors that have recently been shown to be critical for resilience to social stress, physical, and mental health. And furthermore, there's a rich history of them being used as a model species for investigating social reward and motivation-like behaviors. So how do we measure social reward in a laboratory context? Well, perhaps the most popular measure of reward is a condition-based preference test, or CPP. CPP quantifies the reward by measuring time spent in the area associated with the reward, in this case, social interaction. Thus, it relies on the memory of the chamber with the reward, i.e. the social interaction. Thus, I took male and female Syrian hamsters. Thus, I took male and female Syrian hamsters and allowed them to free roam between the 2 different chambers. I determined initial preference. Then I paired them with a same-sex matched smaller stemless hamster and their initially non-preferred chamber, and then as a control, they were paired alone in their initially preferred chamber. And then on the final day of testing, I then measured time spent in the chamber, associated with the social interaction as my measure of social reward. So the results. So both males and females display an increase in the time spent in the social interaction paired chamber. However, the degree of increase was twice as large in females compared to males, thus suggesting that at least in the CPP test, females were finding social interaction more rewarding compared to males. However, CPP is somewhat limited in the assessment of social reward due to its strong memory component. Thus, our lab has recently designed and validated a novel operant social preference task that has less dependent on memory and tests the reinforcing properties of social interaction. Furthermore, it also allows the subject the choice to either engage or not in social interaction. So in brief, if you look over on the right here, there's a main chamber and two smaller adjacent chambers. And the stimulus hamster, which is on the top, sorry, the subject hamster, which is on the top, can access either the left or the right smaller chambers in which the stimulus hamster is confined. And access to the smaller adjacent chambers can be accessed through a one-way entry vertical swing door. And additionally, there are buckets on the back of the door that can be used to serve to hold weights, which I can then manipulate the behavioral cost requirements to access the reward. So here's a video of a subject's first time inside the apparatus. And so then just, no one's surprised by this, they're not on speed. The video's been sped up to 8x for time constraints. But basically, so the subject is on the top in the main chamber, and the stimulus hamster is combined to either left or the right smaller chambers. The empty chamber serves as a locomotive control chamber. Now, notice all the exploratory behavior in the beginning. Now, I want to emphasize again, this is a subject's first time inside this chamber. So they have no previous experience with either the size or the shape of the arena, the apparatus. They have no previous experience with the vertical swing doors or what's on the other side of the doors. However, you notice that as the time goes on, eventually the subject makes its way, is able to, through olfactory communication, sense the stimulus hamster. And additionally, the experimenters were allowed to manipulate and control the duration of the time, that they spend inside the apparatus. In this case, they're allowed only 20 seconds, but we can manipulate the duration of the interaction as a way to also manipulate the reward value. Furthermore, notice the change in latencies from the time that it takes for their first entry versus their second, and then with further entries. So you notice that basically, as the time goes on, they're able to perform this task very rapidly, showing that this operant task is rapidly acquired, and that the social stimulus serves as a powerful motivator. I think I messed it up. I think I broke it. So we'll just do this. Okay. And so, however, what's the effect of adding weight to the door? So in this case, the weights on the door are 624 grams, or 22 ounces. Now, as a frame of reference, an adult Syrian hamster, both males and females, weigh between 130 and 150 grams. So the weight on the door is significantly heavier than their own body weight. And notice how much time is spent attempted by the subject hamster attempting to push open the door. So I can measure the duration of the time spent attempting to push open the door as a complementary measure of motivation. Now, I have performed numerous experiments in which I manipulate both the social reward value, i.e. the duration of the time they spend in the social interaction period chamber, and the behavioral cost requirements, the weights on the door, on the reinforcing properties of social interaction. For example, increasing the time spent in the social interaction period chamber actually decreases the rate of seeking out the social stimulus, and also increases the latency to re-enter the social chamber, thus supporting the theory that the rewarding properties of social interaction may be titrated out and dose-dependent, similar to many drugs of abuse. So are there sex differences in this opposite social preference task? Yes. As you can see here, so on the y-axis is social preference score, which is the number of entries in the social chamber minus the number of entries in the empty chamber. And on the x-axis is the effect of progressively increasing the weight on the door. And as you can see, females are the black and males are the gray, that across various behavioral cost requirements, females are again displaying a greater social preference score compared to males, similar to what we saw in the CPP test. And additionally, if you look at the effect of adding weights to the door, females are also spending more time attempting to push the door open compared to males. Females are the black circles, while males are the squares. Now, there may be many potential explanations for these observed sex differences. However, our lab is theorizing this may be due to females being more sensitive to the rewarding effects of social interaction compared to males. There's a large body of literature supporting the fact that females are more sensitive to various other rewards, including drugs of abuse. Thus, we theorize that if you look at the graph over there on the bottom right, females are basically, this is a graph that looking at social reward value on the y-axis, and on the x is social interaction stimulation. Thus, if females are more sensitive, they're shifted to the left of the curve, the black line, while males are the dotted line. And as you can see, at the level of social interaction that is maximally rewarding for females is only suboptimally rewarding for males. So given these observed behavioral sex differences, I was interested in what may be some of the neural mechanisms that's contributing to observed sex differences. Well, the mesosomal dopamine system, which primarily consists of dopamine-containing neurons in the VTA that project the nucleus accumbens, is the most well-known circuit involved in reward processing. And surprisingly, there are very well-known and characterized sex differences in the mesosomal dopamine system. For example, electrical stimulation of medial forebrain fibers, which are basically the oxytocin, which are basically the dopamine projections to the nucleus accumbens, results in greater dopamine efflux the nucleus accumbens in females compared to males. Females are the closed circles, while males are the open circles. And also, IP administration of an amphetamine also results in greater dopamine efflux in the accumbens in females compared to males, the graph on the right. And finally, females are also more sensitive to the rewarding effects of cocaine. Females display a conditional place preference require fewer training sessions compared to males. And furthermore, they display a conditional place preference for cocaine under a lower concentration compared to males. And very interestingly, if you look at the very far, this bottom graph here for the males, a high concentration of cocaine that induces a place preference in males actually then results in an elimination of a place preference in females, while they do show a cocaine preference under a lower concentration. So given these observed sex differences in the mesosomal dopamine system, I was then interested what system may be driving or regulating the sex difference. Well, very recently, the oxytocin system has been implicated as a critical component in regulating the mesosomal dopamine system. For example, oxytocin receptors are co-localized with dopamine neurons in the VTA. And as expected, injections of oxytocin in the caudal VTA results in dopamine efflux in the accumbens, and this is regulated through the oxytocin receptor. And furthermore, our lab, Elliot Aubrey's lab, has recently shown that oxytocin receptors in the VTA are necessary for social reward in male Syrian hamsters. And furthermore, the Malinka lab has shown that oxytocin-containing neurons in the PVN projecting to the VTA are necessary for social reward in male mice. However, all these studies, as I just said, were only carried out in males. Thus, to investigate if the oxytocin system is recruited during same-sex social interactions in females, I took male and female Syrian hamsters and allowed them to interact in a 10-minute, allow them to socially interact for 10 minutes. Males with males, females with females. Then I measured CFOS colocalization in both the PVN and SON with oxytocin neuronal activity. And as you can see here, both males and females display an increase in CFOS colocalization with oxytocin containing neurons in the PVN. And likewise, so does the SON. Thus, in both males and females, the PVN and SON display changes in neural activity and to similar degrees for same-sex social interactions. However, to test if the oxytocin system is necessary for social reward in females, I took male and female Syrian hamsters and had them again interact in the conditional place preference test. However, prior to the social interaction training sessions, they were treated with either saline or an oxytocin receptor antagonist. And so the red bars are with the saline injections, and as previously shown, both males and females display an increase in the time spent in the social interaction paired chambers with just saline injections. However, the degree of increase, again, was greater in females compared to males. Now, interestingly, injections of an oxytocin receptor antagonist decreases social reward in both males and females, and to similar degrees. Finally, I asked, maybe there's sex differences in the effects of exogenously stimulating the oxytocin system on social reward processing. Thus, as previously, as in the previous study, I took males and females, and this time they were injected with either oxytocin or a highly selective oxytocin receptor agonist into the coddle VTA prior to the social interaction conditioning sessions. Now, as expected, males over there on the right are displaying an increase in the time spent in the social interaction paired chambers with either injections of oxytocin or the oxytocin receptor agonist. However, very interestingly, we get the opposite effect in females, where in this case, injections of oxytocin or an oxytocin receptor agonist into the coddle VTA actually decrease time spent in the social interaction paired chamber, displaying that they're showing a decrease in social reward. So while their inhibition of the oxytocin system is inhibiting social reward in both males and females, exogenously stimulating the system prior to social interaction is having opposite effects, where it increases social reward in males but decreases social reward in females. So this is the same graph here. Now, very interestingly, work from James Rilling's lab at Emory has found remarkably similar results. Over at Emory, he takes men and women participants and allows them to engage in same-sex social interactions, the prisoner's dilemma. And then he measures fMRI activity, bold response, in certain brain regions, in this case the caudate putamen, and then looks at the effects of an intranasal oxytocin treatment prior to these interactions on the fMRI activity. Now, very interestingly, he's found that intranasal at baseline, females again are displaying greater greater bold response in the caudate putamen, which is analogous to the ventral striatum, part of the reward circuit, in women compared to men. Now, very interestingly, intranasal oxytocin treatment prior to these social interactions is then decreasing the bold signal in women, but is then also increasing, and has the opposite effect, increases the bold signal in men. Thus, these data altogether fit nicely with the hypothesized inverted U-shaped model of social reward value, where females are shifted to the left of the curve. Thus, if I can just show you guys, so at baseline here, with no drug treatment, females are at the maximum reward value for social interaction, for social reward value, while men are at the suboptimal level. However, inhibition of the system, treatment with an oxytocin receptor antagonist, decreases social reward in both women and men. However, exogenous stimulation of this reward system, treatment with an oxytocin and oxytocin receptor agonist, pushes females past the maximum reward value, well, now it becomes less rewarding, aversive. While men are pushed to their optimal reward value, well, now it becomes more rewarding. Thus, to summarize, female hamsters are displaying greater social preference score compared to males in both the CBP and OSP behavioral tests. The OT system is necessary for social reward in both males and females. And exogenous treatment of OT and or activation of OTRs in the VTA can decrease social reward in females, but can increase social reward in males. Both human and animal studies alike. Thus, there may be a sex-dependent inverted U dose-response relationship between reward value and social interaction duration, OT activation. And then just to bring everything back, this may also fit nicely with the observed sex differences in the prevalence and predisposition for various psychiatric disorders such as autism spectrum disorder, where females are more sensitive to social interaction, social reward value, while males are more sensitive or prone to deficits in social reward. And so I'd like to thank you all for being here today, listening to me talk. I'd like to thank all the undergrads, fellow grad students, postdocs, technicians, and the funding provided by the NIH.
So we have several minutes for questions.
Yes, David. Yeah, no, that's a great question. Yeah, so David was curious about the effects of their social housing. So all my hamsters are single housed at least 3 to 4 weeks prior to this testing, and that part of that definitely builds up part of their social motivation. However, it would be really interesting to see, if there might be sex differences or just housing differences in terms of whether they're group housed or isolated in terms of their social motivation. Yes, Nate. So Syrian hamsters display very robust and highly ritualized dominant-subordinate relationships. Where basically you put two of them together, one will be display a little bit of aggression, and then the other one becomes very submissive. And so some male mice, and there's obviously differences in strain and breed, but similar behaviors are also seen in terms of the conditioned to feet model. However, it's really nice. The main reason why we use Syrian hamsters is because, unlike other rodents, both males and females display the same quality of social behavior. Yeah, Manfred. Hmm. Yeah. Yes. 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 Hmm? Yeah. Yeah. So yeah, there's a lot in terms of like, obviously the quality of the social interaction has a big effect on whether it's rewarding or not. And I would obviously expect that, yeah, the dominant is going to find it more rewarding than the one that's being submissive. And I haven't carried out those studies to look at the differences in terms of just time constraint. Would be very interesting to see that, yeah, just, and with the Syrian hamsters, I could definitely design a model where to see, will they still, push through the door or spend more time in the chamber associated with being submissive. Yeah. Yeah. So when I do these studies, by actually manipulating their social housing, I'm able to then make sure that my subject's the dominant, and my stillness is always a submissive. So my stillness hamster is always group housed, which lowers their aggression levels, while my subject is single housed, and that increases their aggression levels.
Time for one more. Laura?
Mhm. Yes, great question. So generally females are a little bit heavier than the males. However, the difference is about no more than like 10 grams.
Thank you, Jonathan.
Yep, thank you.
All right, our next speaker is Sabrina Na from Tricia King's lab in psychology. And Sabrina is going to tell us about white matter network topology, relates to cognitive flexibility and cumulative neurological risk in adult survivors of pediatric brain tumor.
Still your slides. Well, thank you so much for that introduction. So today I'm going to be presenting my dissertation research where I use graph theory methods to explore white matter network topology in adult survivors of pediatric brain tumors. So just as a brief overview of the literature in this group, cancers of the brain and central nervous system are the second most prevalent type of cancers in children. And recent improvements in survival rates have led to an increased research emphasis on the quality of survival as more and more of these survivors reach adulthood. Research suggests a myriad of late effects in this group, and this includes deficits in many areas. That includes cognitive deficits and also in the domain of executive functioning. So executive functions relate to a group of higher order cognitive skills that are used for independent, purposeful, and goal-directed behaviors. So this is still kind of an early area for neuroimaging, but the neuroimaging studies in this group have established both macrostructural and microstructural effects on the brain. And this includes lower white matter integrity overall and in specific tracts of the brain. And these studies have also found that the level of white matter integrity relates to behavioral outcomes, as well as risk factors such as higher treatment intensity or the presence of radiation treatment. So these studies together suggest that changes in white matter may represent the neurobiological underpinnings of some of these outcomes. So these studies so far have identified specific regions of the brain that are different between survivors and controls. They really rely on the assumption that specific regions of the brain are responsible for specific functions. But these studies have yet to employ analyses that investigate how distal brain regions are working together in a unified system. So one way to investigate system-level properties is by using graph theory. So this method is particularly helpful when you're examining higher-order behaviors that depend on the integration of information from spatially distributed regions of the brain, just like executive functions. So graph theory is a branch of mathematics, and it provides quantitative metrics to describe the properties of complex graphs. So graph theory has actually been around for a long time, and it's a branch of mathematics that was first developed by the mathematician Euler in 1736. So at that time, the city of Königsberg in Prussia was set on both sides of a river, and it included 2 large islands connected to each other and to the 2 mainland portions of the city by 7 bridges. So Euler's question at the time was to ask, is it possible to walk through the city and cross each of these bridges only once? So his attempt to answer this question in a systematic fashion laid the foundations of graph theory. So his approach involves distilling a complicated system into smaller components, or a graph made of nodes and edges. So here you can see a simplified graph that kind of retains the main portions of the land and the bridges. And then you can simplify this even further. So the land masses are replaced by nodes, and then each bridge is modeled as an edge. So that's indicated by a line. So this resulting mathematical structure is called a graph, and it incorporates information about all of the land regions as well as the bridges from the more complex system into a much simpler graph. So over the past decade or so, this method has been applied to the brain to understand how distal brain regions are working together in a unified system. So when using graph theory, the researcher defines how to divide up the brain into smaller components. So they can decide which regions they want as nodes, how many regions that they want. And then the researcher also can use different network construction tools, such as tractography or functional connectivity, to estimate the connections or the edges between each brain region pair. And then we can derive quantitative metrics of the network. And there are 3 types of measures that we can glean from the network overall. So the first type are measures of segregation. So this describes the extent to which information is processed locally in a small region. And I'll provide examples of what these metrics look like and how they're calculated in a couple of slides. The second type are measures of integration. So these describe the extent to which information is processed across spatially distributed regions. And healthy adults demonstrate a balance of both segregation and integration in their brain networks. So both features are essential, and they're consistent with the idea that specific regions of the brain are responsible for certain functions, but that also that the brain works as an integrated system of parts. There are also measures of centrality that measure how important any particular node is to the network. So the nodes in a healthy brain follow a power law distribution. So if we plot a histogram of all of the nodes in the brain, based on how many connections they have to other nodes, it may look something like this graph. So the vast majority of nodes in the network have a low number of connections to other nodes, but there are several nodes that have an exceptionally high number of connections to other nodes, and these regions are essential for integrative processing. And these highly connected nodes are defined as hubs. And so they have the highest measures of centrality in the entire network. So these regions are often found in heteromodal cortex, or cortical regions that receive input from many regions. So these regions include the bilateral precuneus, superior frontal gyrus, superior parietal cortex, hippocampus, putamen, and thalamus, among other regions. And these are commonly implicated as having very strong importance to brain networks. So this type of network setup overall ensures resilience to random attacks, but also provides a way for information to flow and travel quickly and efficiently across the entire system, all while reducing biological costs. So there have been studies that utilize graph theory for various neurological disorders, such as multiple sclerosis, epilepsy, stroke, and traumatic brain injury. And all of these studies suggest that these disorders are associated with disruptions in the network. And chronic stages of the injury in particular are associated with reductions in all of these measures of integration, segregation, and centrality. Research also suggests that hubs are disproportionately and consistently affected across many disorders. and many clinical populations, and they all exhibit significant decreases in their measures of centrality in chronic stages of illness. And finally, the metrics that describe the integrity of these networks, of these brain networks, correlate significantly with complex behavioral outcomes, and the magnitude of impairment is related to the integrity of these brain networks. So the purpose of this study was to use graph theory methods to examine structural brain networks of adult survivors of childhood brain tumors. And the first aim was to determine whether there are alterations in structural networks in adult survivors compared to healthy same-age peers. And the second aim was to determine whether these metrics actually relate to behavior and underlie cognitive flexibility performance. And cognitive flexibility is a subdomain of executive functions. So to briefly describe my sample, I had 38 survivors, all of whom were adults. These survivors were an average of 14 years post-diagnosis, and so this suggests that this represents a really specialized group of very long-term survivors of brain tumors. And we also recruited a healthy control group who were matched for age, gender, and socioeconomic status. We also acquired diffusion-weighted data on a 3T Siemens Trio MRI scanner with 30 tensor directions. So diffusion tensor imaging measures the rate and direction of water diffusion, and this data can be used to provide estimates of white matter integrity in white matter tracks. And then we can use tractography methods to reconstruct 3D maps of white matter pathways in the brain based on mathematical algorithms. I also used a parcellation scheme that divides the brain into 120 different regions, or nodes. And then I calculated the average white matter integrity, or FA, of all of the voxels along the streamlines that linked two nodes or any particular two brain regions, and they were defined as edges in my network. For measures of behavior, I used cognitive flexibility as my domain of interest, and this was measured using a modified Stroop task. So participants had to switch between reading the color that a word is printed in and reading the word itself based on a rule. And scores were age-normed. I also used the neurological predictor scale score as a measure of cumulative treatment and neurological risk. So this scale incorporates common treatments for brain tumors such as radiotherapy, chemotherapy, and also the presence of common neurological conditions like hydrocephalus. So these all serve as risk factors, and higher scores on this scale indicate having more of these risk factors and have been associated with poorer cognitive intelligence and adaptive outcomes. So I'll just briefly discuss the metrics that were extracted from the network. So for the measures of integration, I used global efficiency. So efficiency is the inverse of the fewest number of edges between two nodes. So to travel from node 1 up in the upper left corner and node 2 in the bottom right corner, the fastest way to get there is with 3 steps or 3 edges. So that's the path length, 3, and the efficiency is the inverse, so that's 1/3. So a high level of global efficiency suggests a high capacity for parallel processing and a more efficient network. So you get an average of all of the global efficiencies across all of the node pairs in the network. So for a measure of segregation, I use the clustering coefficient, and this measures the probability that the neighbors of a node are also connected to each other in the form of a triangle. So for this example, for node 1, which is indicated in yellow in the center there, the clustering coefficient is 1/6. So for all of the neighboring nodes, there's only one triangle, or one of the neighboring nodes are connected to each other in red. But there's a possibility of 6 different edges that are possible in the form of a triangle. So the clustering coefficient for this node is 1/6. So higher levels of clustering coefficient is thought to measure higher levels of local processing. And you can average the clustering coefficient of all of the nodes in the network as an estimate of how well information is processed within small regions. So another measure of segregation I used was modularity. So this measures the level to which there are communities that have more connections with one another than is expected in a random null model. Higher levels of modularity suggest higher levels of segregation in networks and suggest that there are more communities with specialized functions. And then for a measure of centrality, I used betweenist centrality. So this is a property of an individual node, and it provides a quantitative description of how important that particular node is in the network. So betweenist centrality measures the number of shortest paths that must pass through one particular node. So in this series of networks, the yellow node in the far right has the highest betweenist centrality of all of the nodes in other networks, because all of the nodes have to pass through that yellow node in order to get to any other node in that network. So a high betweenist centrality suggests that that node is highly important to processing in that network. And so I wanted to use a preferential damage measure, and there's something called the Hub Disruption Index. And I won't go into the mathematical details, but basically a significant negative slope to this graph indicates more preferential damage to hubs. All right, so just to go over the results, the purpose of the first aim was to determine whether there are any alterations in structural networks when comparing between the two groups. And here you see that global efficiency and average clustering coefficients are lower in survivors compared to controls. So this suggests that there is an impact to how well information is processed both locally and globally in survivors. There's also evidence for preferential damage to hub regions, or the specific regions that are essential for integrative processing in healthy brains. So here we have a graph of a one-to-one mapping of all of the edges in survivors on the x-axis and healthy controls on the y-axis. So perfect linear correlation is represented as the dark black line. And you can see that in areas of medium to high white matter integrity, as indicated by the two green arrows, there are more dots above the black line than under it, whereas in areas of low FA, as seen in the lower left corner, there's about an equal smattering of dots above and below the black line. So this really indicates that there is a preferential damage to tracks that have higher white matter integrity in survivors, and these are consistent with information highways in the brain or long-distance track connections. And here we have a graph of all of the hub disruption indices for all of the survivors. And you can clearly see this steep downward slope for all of the survivors, which indicates preferential damage to hub regions. And here we have scatter plots relating neurological risk and properties of the white matter network. So lower levels of global efficiency and lower levels of average clustering coefficients are both significantly associated with higher levels of neurological risk. So if that survivor had radiation, chemotherapy, and other kinds of neurological risk factors, that was associated with more alterations in the white matter network. So the second aim was to determine whether properties of the network are related to behavior, specifically performance on tasks measuring cognitive flexibility. So here, both global efficiency and average clustering coefficients were correlated with performance on the cognitive flexibility task. So if you have more alterations to your network, that's associated with poorer performance on the task. And additional mediation analyses showed that global efficiency of the white matter network mediates the performance differences in cognitive flexibility between the two groups. And it also mediates the relationship between neurological risk and cognitive flexibility. So overall, these results are showing that there are alterations in structural networks in adult survivors in the form of reductions in global integration, segregation, and also preferential hub impact. I think this preferential impact is particularly interesting, considering that the sample of survivors that we studied were heterogeneous with regard to tumor type and location. So this suggests that there may be a level of specificity to the regions in the brain that are most at risk across long-term survivors of varying types and locations of brain tumors. And this is also highly consistent with literature from many other clinical populations with neurological disorders where you do see this preferential hub impact. We also saw that cognitive flexibility performance differences between groups were mediated by differences in white matter structural network topology. So I think this suggests that features of these brain networks have power to explain differences in complex behaviors such as cognitive flexibility. So put it all together, brain networks of healthy individuals are associated with a balance of segregation and integration. So this is so that information can be efficiently transferred across and between brain networks while still maintaining low biological costs. And studies of healthy developing brains indicate that brain networks undergo highly dynamic changes from infanthood to late adolescence. And these networks are changing from relatively random configurations to networks that optimize the balance between information segregation and integration. And these changes together support behavioral and cognitive developments. But the rapid changes that are occurring in brain networks during development also render it very vulnerable to neurological insults. And in survivors of pediatric brain tumors, they're experiencing disruptions during these really critical timeframes when structural and functional networks are actively being optimized for efficiency. So the results of this study suggest that these network alterations persist even when survivors have grown into adulthood. And these alterations also represent the neural underpinnings of behavioral outcomes, such as cognitive flexibility. So I think there are a host of future directions at this point. I think that understanding structural topology lays the groundwork for understanding functional network organization because functional networks are shaped and constrained to a certain extent by the underlying structure. I also think that understanding the flexibility and diversity of functional network organization within the constraints of anatomical connectivity can provide really important insights into how the brain repairs, recovers, and functions over time following a neurological insult. There's also literature that suggests that graph theory may have some utility in clinical settings. So metrics derived from structural and functional brain networks have been used as biomarkers in clinical groups, such as patients with temporal lobe epilepsy. So they've been shown to predict patients will have better outcomes after surgery. So I think that graph theory could be used to guide neurosurgical planning to avoid neurological deficit, predict the efficacy of treatments, and also to identify the patients at risk for poorest outcomes. So this study was cross-sectional and couldn't provide information about causation, so I think that longitudinal studies will be crucial to establish timeframes and causes of change. And I also think that this work will be necessary in larger samples to establish that graph theory metrics do have clinical utility in survivors. But I think that the findings from this study provide a really exciting window into the possibility of using this method to predict long-term outcomes in personalized medicine for survivors of brain tumor. So before I end my presentation, I'd like to thank all of the funding agencies and the individuals that made this research possible, specifically Dr. Tricia King, my advisor, all of the members of my dissertation committee, the participants of the study and their families without whom we would not be able to do this work, and all the graduate students and research assistants involved in this lab. Thank you all for listening.
So we have time for a couple of questions, and if you would, and if you can make your way to the mic, it would be best for people to hear the question. Or speak loudly.
Hi, here.
Hi.
I'm Yang from chemistry and CDT. So great talk. Have you looked into your hotspot or connections correlated with molecular biomarker, which now the information is available?
I have not, but it would be really exciting collaboration, I think.
Just a point to see that we have applied graph theory for use for predicted calcium networks.
Yeah.
Chris?
That's a really good question. So for when they were diagnosed, it ranged between, I think, 10 months to 17 years. So it was a really large range. I actually did test whether age at diagnosis and time since diagnosis was associated with any of these metrics, and they weren't. But I do suspect that there may be a moderation. So if you had more severe treatments like radiation, chemotherapy, and usually they co-occur, then I think that you will see more of this relationship between younger age at diagnosis and outcomes. Yes. Yeah, I mean, we would need to collect, I think, very specific diffusion tensor imaging data and have a lot more advanced algorithms in order to do more directional approaches. But yes, I think that would be fascinating to do.
Thank you, Sabrina.
Thank you.
All right, our next speaker is Yang. You go by Dawn, right? Wu from Chen Jiang's lab in biology, and she is going to tell us about hyperexcitability of medullary respiratory neurons, irresponsible for breathing abnormalities caused by MECP2 disruption.
Hi everyone, my name is Yang. I'm from Dao Zhang's lab, and today I'm going to give a talk about Rett syndrome. The title is Hyperexcitability of a medullary respiratory neuron is responsible for breathing abnormality caused by MECP2 disruption. So Rett syndrome is a severe and progressive neurological disorders. 90% of the disease can caused by the mutations in a gene called MECP2, and this gene is located on X chromosome. The disease is predominantly affected the female baby girls, with an incidence of 1 to 10,000 female births. And the people with Rett syndrome usually suffers from characteristic breathing disorders with a high incidence of sudden deaths. And those breathing disorders usually include the apnea, shallow swallowing, breath hold, and the L swallowing. So for a long period, for a long period, the study of MECP2 knockout mouse is the major model used in the Rett syndrome study. But the size is smaller. But recently, a novel MECP2 knockout rat is available, and the data allowed us to perform deeper studies looking at the mechanism of these breathing disorders. We observed we observed the breathing abnormalities under two conditions. One is using the plethysmograph on the awake animal. Another is using the phrenic nerve recording test on the anesthetic animal. And we find that the MECP2 knockout rat has the high apnea rate and the irregular breathing. And when the animal is reaching the human endpoint, usually is when the animal is dying, those breathing disorders will become severe. We still have a lot of open questions, such as what type of respiratory neurons are defective regarding their firing pattern and overall activities. Are there any changes in cell grouping, and what really happens during the apnea? What causes the apnea? And does phrenic nerve activities completely cease during apnea? Is it possible there is a general neuronal process contributing to the breathing disorders? If so, can we use certain pharmacological interventions to correct the problems? To answer these questions, we performed the in vivo electrophysiology, directly looking what is happening on those respiratory neurons in brainstem. So in brainstem, there are many respiratory neurons distributed in different locations. There are two major locations. One is located in the dorsal area, one is located in the ventral area. And probably 80% of the neurons is located in the ventral areas. So the column we are going to study is called a ventral respiratory column. And that contains three groups. One is inspiratory group, which is in the middle, contain pre-body syndrome complex, and RVRG group. And another two is expiratory neurons, which contains in the Bode-C complex and the RVRG group. So basically, the breathing can be separated into three phases. One is the inspiratory phase, which is indicated by 1, and another is post-I phase. We call it early expiration, indicated by number 2. And the third one is the late expiratory phase. We also call it E2 phase. And usually, those respiratory neurons only firing during one of the phase. We use the phrenic nerve discharge as the references because usually the phrenic nerve only firing during inspiratory phase and maybe partially in the post-I phase. So PN is the phrenic nerve discharge. So in the electrophysiology studies, we have the chance to looking closely what really happens on these respiratory neurons. And we find the firing activities actually changed in the MECP2 knockout animal. Taking an example, we totally look at three groups. Figure A indicated the inspiratory neurons, RVRG. B and C are both expiratory neurons, CVRG and Bode-C. Taking RVRG as an example, if you're looking closely, you can see the firing activity of the neuron actually extended their duration. The firing duration of the RVRG actually is longer compared with wild type. But the frequency is relatively lower. So to looking at their overall activity, we use two parameters. One is indicated in figure G. It is the spike number during each burst. And another parameter is the activity. We use as a frequency, firing frequency times the duration to represent the overall activity. So both spikes number during each burst and the overall activity, they increased in MECP2 knockout group, which indicated by figure G and H. The expiratory group, we also find the similar results. So both of the inspiratory group and the expiratory neurons, they have a higher overall firing activity. We also compare their locations and or distributions of those cells. We find there is no clear evidence indicated there has certain changes on the location. You still can see in the middle, the yellow color indicated the inspiratory cells group. And the blue color, one is located in rostral, one is located in caudal. One is Bode-C complex, another is CVRG complex, indicated by blue color. They still maintain certain locations. The green color actually is the overlapping of both inspiration and expiration neurons. So between Y-type and MECP2 knockout, there's no clear differences. And the firing pattern is also changed in both inspiratory and expiratory neurons in MECP2 knockout animals, comparing with Y-type. So figure A and B, we're looking at a special group of neurons. They firing the face-bending cell patterns. Their firing activity will cross both expiration and inspiration. And those group of neurons, the ratio is more in MECP2 knockout animals comparing to Y-type, which is indicated by figure C. And in figure D, you can see the firing duration in E phase, expiratory phase, is longer compared to Y-type. Another piece of evidence indicating the firing pattern changing is found in post-I cells in Bode-C complex. Both Y-type and MECP2 knockout animal will have these post-I cells. We find the ratio of post-I cells in MECP2 knockout animal is shorter, it's smaller comparing with Y-type, indicated by figure F. And probably this is also can be explained by hyperexcitability of the neurons, because when the cell is hyperexcited, the post-I cell will extend their firing activity to the E2 phase, the late expiratory phase, and mix them with the other type of expiratory cells. Besides this, we're also interested about what really happened during apnea or what caused the apneas. Before we performed the studies, many people believe that the cause of apnea is caused by the suspension of the central inspiratory activities. But after we performed the study, we find the opposite way. We compiled all the both inspiratory and expiratory cell firing activities during apneas. We find during apnea, actually the inspiratory cell and the face-bending cell, they remain their activities. Figure A and B indicated that. Figure A is the inspiratory cell, B is the face-bending cells. During apnea, which indicated by the arrow bar, the firing activity remains. So that probably suggests that the cause of apnea is caused by the defect of EI switch rather than the suspension of the inspiratory activities. Another piece of evidence indicated this is finding from the abnormal phrenic nerve discharge, which is indicated by the bars, the two arrow bars. We find that the phrenic nerve will have some ectopic discharge. What is called ectopic discharge is because when apnea happened all during expiration, we believe the phrenic nerve are supposed to be silenced. But in our recording, you still can see some fuzzy stuffs in the second line of the recording. That is a phrenic nerve discharge. They're still firing their activities. So those data suggest probably in MECP2 knockout animals, those neurons, respiratory neurons, they are going through the hyperexcitability stage. And I have one slide. This one is indicated that those face-bending cells, some of those face-bending cells can actually project to the spinal cord, contributing to the formation of ectopic signals. So the hyperexcitability seems a common state in the MC2 knockout animal. The reason causes, one hypothesis is probably caused by the defect of inhibitory input. Maybe it's caused by GABA input. And if we, to test this hypothesis, if we apply the GABA augmentation reagent, maybe it can correct the problems. We have two evidence indicating this. One is found when we apply a drug called NNC711. It is chemical. It is a GABA reuptake blockers. It can augment the GABA input. We apply the drug using the IP injection, and we have the chance to record the long period, long period recording in neurons, face-bending neurons specifically, indicated in figure 8. On these neurons, we find after we apply the drug, just 20 minutes later, it will change in the face-bending cells back to the normal inspiratory pattern. And it will remain, last longer for during the rest of the recording. Not only it can change the phrenic nerve face-bending neurons back to the inspiratory phase, it also can reduce the ectopic discharge we find on the phrenic nerve recordings, which is indicated by figure A on the right side. So in the small box, you can see after we apply the NNC711, the ectopic signals of a phrenic nerve discharge reduced. We also find another piece of evidence, such as when we augment the GABA system as a symptom, the abnormal disorders actually reduced. Another reagent we use is called a THIP. It is a GABA, sorry, I put the wrong name here. It is an extracellular synaptic GABA A receptor agonist. So when we apply the THIP in figure B, you can see just 40 minutes, just about 1 hour after we apply the drug, the breathing patterns become relatively normal, even similar as a wild type. And it also reduces ectopic signals. So the THIP also have a rescue effect. We selected the animal which they already reached the human endpoint. So when the animal is dying, usually they suffered from the severe breathing disorders indicated by figure A. And on their face, around the eyes and the nose, you can find the red tear, the bloody clusters all around their surface. This is because the animal is undergoing the stress or the sickness. So when the animal is reaching the human endpoint, we give the injection, IP injection again. Then we find just 1.5 hours, the breathing is become relatively normal. Reduce the apnea rate, changing the irregular breathing to the regular breathing. And the second day, when we look at the animal is become, I couldn't say completely healthy, but 90% recovered. Figure F is a picture I took after the injection, 1 day after the injection. You can see the red clusters around the nose and the eyes reduced. The animal's condition become better. And the third day, you can see after 48 hours, those bloody things was cleaned by the animal itself. I have a video can better show this effect. So when the animal is reaching the human endpoint, you can see there is a lot of bloody things on their surface, on their face, and there is a lot of apnea and irregular breathings. But just 1 day after the injection of THIP, the conditions become better. So this is the recording we did after 1 day of the injection. Yes. Okay. So the animals become relatively normal comparing to the conditions. And this animal is rescued by the 1-dose THIP injection. So we also took a video about 4 days after the injection. The animal moves freely in the cage. It looks happy, and it didn't seems it's stressful. If the animal can be rescued, we also give it a 1-week THIP oral treatment in their drinking waters, and this is a recording after 23 days. So before this animal is almost dying, but single dose can rescue its life, and a 1-week treatment can extend the animal life almost longer than 3 weeks. So this is a really exciting results. So from our studies, we observed that the neuronal hyperexcitability is rather common among all major types of respiratory neurons. And apneas in the neurons seems result from the defects in the functional expiration-inspiration switch, rather than the suspension of central inspiratory activities. Augmentation of the GABA synaptic inhibitions reduce the abnormal neuronal firing, resume the MECP2 breathing pattern to the wild-type pattern, and rescue most of the animals reaching the human endpoint. So thank you all. I should thank my PI, Dr. Zhang, and my committee, and all my lab mates who helped me to perform this study. So any questions? Questions?
Will?
This inhibition amount is happening. We are studying the ventral respiratory columns, which located in the brainstem. So that specific area is very close to the facial nucleus. So it's more caudal to the facial nucleus. So in that group, 90% of the respiratory cells is located over there. So we believe probably this defect is a general condition.
Very great talk. And have you looked into calcium? Because there's quite a bit of publication shows that THIP indeed alter the calcium signaling dynamics.
We didn't look the molecular level. We only look at what really happens on those neurons' activities. So we didn't have a chance to do this. But maybe in the future, we will perform certain studies.
Angie?
Yes, we did some studies on the long-term treatment, but not in the rat model. We did the treatment in the mouse model, and we find the expression level. We probably couldn't change the expression level, but we did, in fact, a lot of synaptic signals. So the long-term treatment is also, you can find the published paper in our website if you are interested.
All right. Thank you very much. So we now have a break until 2:55. If you could come back about 2:50, that'd be great.